Hi, I'm Joseph Chen, and I'm working in the Smith Research Group with Bianca, Philip, and Nico. I'm a current senior at Phillips Exeter Academy, located an hour north of Boston. On campus, I'm heavily involved in environmental activism and sustainability efforts. Having worked in the lab with some wonderful people, I'm now excited to share with you all our research project. Antibiotic resistance is a growing threat to public health and the environment. With the spread and evolution of antibiotic resistance, healthcare costs have risen, stronger diseases have emerged, and mortality rates have increased. Wastewater treatment plants, which act as gateways to the environment, are believed to be hotbeds for antibiotic resistance proliferation. The nutrients and antibiotics in wastewater effluent, combined with a plethora of bacteria, allow for propagation of antibiotic-resistant genes within wastewater microbial communities. However, if we better understand the effects of antibiotic loading at wastewater treatment plants on microbial community structure, we can mitigate and avoid the proliferation of antibiotic-resistant genes into the environment. Despite heavy research about antibiotic loading in wastewater treatment systems, little is known about the resulting bacterial communities, especially in emerging biotechnologies such as anaerobic membrane bioreactors, or AnMBR for short. Therefore, our research project delves into the effects of antibiotic loading on bacterial communities within AnMBRs. Elucidating the effects of antibiotic loading and bacterial community structure will inform the resilience and susceptibility of certain bacterial taxonomies to antibiotics. This, in turn, enables the creation of a better represented database which could be referenced by other environmental samples subjected to antibiotic loading. Even so, future work that correlates antibiotic-resistant genes with resilient bacterial communities can inform the likelihood of certain bacterial hosts to the antibiotic-resistant genes. To conduct our research, an AnMBR was operated under two conditions, one with antibiotic loading and one without. DNA extracts were taken at both conditions from the biosolid and effluent. Through polymerase chain reactions, primers targeted the V4 region of the 16S rRNA for amplifications. Illumina MiSeq was then used to sequence the amplicons. Afterwards, the bioinformatics software Mother processed the sequencing data. By cleaning the data of errors caused by alignment and sequencing, clustering the sequences by 97% similarities to unique OTUs, and classifying the OTUs based on the reference database Silva, we were able to analyze the community structures. To visualize the implications of our methods and the results, we use a rarefaction curve to evaluate the sequencing depth of the sequencer used. Similarly, we used relative abundance graphs to better picture the microbial community structures. Lastly, we performed and plotted a principal coordinate analysis plot to illustrate beta diversity of our samples. The rarefaction curve revealed that the effluent samples were better represented than the biosolid samples with Illumina MiSeq. In addition, the biosolid samples had more OTUs than the effluent samples, implying greater richness. The relative abundance graph by phyla revealed that the effluent samples had higher relative abundance of protobacteria and lower relative abundance of unclassified bacteria compared to the biosolid samples. In addition, the effluent samples had fewer less abundant phyla. Upon antibiotic loading, the effluent samples experienced a notable decrease in the relative abundance of protobacteria, paired with an increase in relative abundance of unclassified bacteria, less abundant phyla, and OP11 bacteria. Lastly, the PCOA plot showcased that the biosolid community structure was clearly distinct from the effluent community structure. Though the biosolid communities before and after antibiotic loading were similar, the effluent communities before and after antibiotic loading were comparatively different. From our results, we were able to qualitatively determine which phylum was more resistant to antibiotic stress. We also showcased that AnMBRs are capable of retaining high levels of bacteria in the reactor, inferring their capacity to decrease antibiotic resistance loading onto the environment compared to conventional treatment systems. I've always been interested in the intersection of environmental sustainability and technology, as well as curious about research in academia. And through this research experience, I was able to explore and learn more about both of these interests. Now, not only am I more interested in pursuing a major in either environmental engineering or bioengineering, I'm also pivoting my course load to take genetics and marine biology in the spring to further explore those interests. I'm excited to continue down this path, hoping to use my experiences and knowledge to build environmental impact-driven technology in the future. I want to give Bianca Costa and Philip Wing a huge thanks for mentoring me throughout the 7 weeks of the research project, as well as Nico for being such a wonderful research partner. I want to also thank Dr. Mills and the Center mentors for putting in the time and effort to make the experience educational, exciting, and fun. Lastly, I want to thank Professor Smith for inviting me to the research group, and I'm excited to come back next summer and help out in person. Thank you for listening.